Hi, welcome for our series of presentation. In the previous presentation, we were looking at the endoplasmic reticulum, and we saw that we have two basic types of endoplasmic reticulum. That is the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The surface of the endoplasmic reticulum is covered with ribosome sometimes. So when it's covered with ribosome, we call it rough endoplasmic reticulum. When it's covered with ribosomes, we call it rough endoplasmic reticulum. So normally, this is how we see it when we are viewing with an electron microscope. As a protein is being made on these ribosomes, it enters the lumen. The protein enters the lumen. So the lumen is basically the inside surface of the ER, the inside surface of the ER. In the lumen, the protein may be modified by having sugar chains added to it, which can help the protein fold or give it stability. So sugar chains are added to the protein. Sugar chains are added to the protein, and the main function of these are to help the protein fold or give it stability, which help to fold the protein or give it stability. So these foldings, of course, provide many functions because they will give the protein the required structure. So not all ribosomes are bound by ribosome. Uh, in fact, some of the ribosomes are just suspended in the cytoplasm. Some of the ribosomes are suspended in the cytoplasm. So some ribosomes are suspended in the cytoplasm. So some are suspended in the cytoplasm. So proteins made on suspended ribosomes are typically used in chemical reactions. So these ones are typically used in chemical reactions. So proteins which are produced here, they are basically used for chemical reactions. So surfaces of the ER have, that do not contain ribosomes are the ones that we call the smooth endoplasmic reticulum. The smooth endoplasmic reticulum makes lipids and performs a variety of other functions, such as breaking down drugs and alcohol. So the smooth ER makes lipids and also and performing other functions like perform other functions perform other functions like breaking down breaking down chemicals. So it breaks down chemicals such as drugs and alcohol. So if someone does not have this and he consumes these drugs and his smooth ER doesn't function well, the impact will be detrimental. The next that we are going to look at is the Golgi apparatus. The Golgi apparatus is also, it is one of the organelles found in the cell, that is the eukaryotic cell. Generally, the Golgi apparatus, unlike the ER, which normally is uh, is working together with the ribosome, the ER, what has been made by the ER, the proteins which are made by the ER are moved to the Golgi apparatus for maybe for more additional stuff to be added on it. So we mean that here, from the ER, protein generally moves to the Golgi apparatus. So from ER, protein moves to Golgi apparatus. Golgi apparatus. Some people call it Golgi body. So this, what happens here, its membrane contain enzymes. Its membrane contain enzymes that make additional changes to proteins. Its membrane contain enzymes that make additional changes. Changes to the protein. The Golgi apparatus also package protein. So it also package protein. It also package protein. So first of all, it makes additional changes, then it packages them and stores them. So some of the packaged proteins are stored in the Golgi apparatus for later use. Some are transported to other organelles within the cell. Still, others are carried to the membrane and secreted secreted outside the cell. So some may be stored in the Golgi apparatus, some of these proteins, some of the protein is stored in the Golgi apparatus. Some of it will be transported to other parts or other organelles that require it, and some of it will be taken to, out of the cell. It will be removed out of the cell to where it is needed. So this is just but the structure of the Golgi apparatus. The Golgi apparatus modifies, packages, and transports proteins. That is its main function. The next that we are going to look at is the vesicles. So we are going to look at the vesicles. So cells also need to separate reactants for various chemical reactions until it is time for them to be used. So what happens is a, A general name used to describe this small membrane or bound sacs that divide some materials from the rest of the cytoplasm and transport these materials from place to place within the cell are called the vesicles. So vesicles are generally short-lived and they are formed and recycled as needed. So one, they they are formed when needed. And their main function is basically uh, to separate reactants from various chemicals and also to transport materials uh, from one area to another within the cytoplasm. After proteins have been made, part of the ER pinches off to form a vesicle. So part of the ER pinches off, pinches off to form a vesicle. So this vesicle that has been formed, uh, this surrounds the protein. So protected by the vesicle, the protein can be safely transported to the Golgi apparatus. There are there modifications will be made to the protein and it will be packaged inside a new vesicle for storage, for transport or secretion. So we can say that these ones, they act like transport materials. So their work is basically to transport materials or proteins which have been made from one area of the cell to another and also to protect it from maybe being digested by enzymes within the cell. The next part of the cell, or another one that we are going to look at, is the mitochondria. So mitochondria supply energy to the cell. Mitochondria supplies energy to the cell. It supplies energy to the cell. So it is very important. Mitochondria is bean shaped, if we look at it. So it has two membranes. The inner membrane has many folds that generally increase its surface area. So it has two membranes. The inner membrane is has many folds. The inner membrane, sorry, has many folds. So the importance of these folds is basically to increase the surface area. So within these infolds and compartments, a series of chemical reactions take place that convert molecules from food you eat into usable energy. So we can say that inside these inner molecules, this is where respiration really takes place, or the conversion of, food molecules into usable energy. So unlike most organelles, mitochondria has only ribosomes and DNA. So it has, mitochondria have their own. Mitochondria have their own, this is unique, their own ribosomes. They have their own ribosomes and DNA. So this is entirely unique to mitochondria and some other organelles. So because of this fact, scientists believe that originally they used to be free living prokaryotes. They used to be free living prokaryotes. Prokaryotes. So scientists believe that in fact mitochondria used to be cells, that is eukaryotic cells, that were taken into other cells which are larger in size. So this relationship must have helped both organisms to survive. So this is like a symbiotic relationship. The next organelle that we will talk about is the vacuole. Is the vacuole. The vacuole, so we will talk about it in the next video. We will stop here for this. And in the next video, we will be talking about the vacuole and its functions.